this, and trans-lactose starch working. Okay? Now here is something we also want to mention. What are those enzymes? What are they really for? This, we need to talk about something else, which is you need to know the structure. Okay, what is the structure? This is a huge bacterial cell. Let's say, okay, I have a lactose. Okay, what is the lactose? Lactose is a disaccharide, double sugar we call. What it looks like? They are composed by a very two common monosaccharides, which is glucose and glycosidase. So CH2, OH, and here we also want to draw the same thing. Only one place is different. Okay, OH, H, OH, H, OH, H goes here, you go OH is up, H is down, H, CH2, OH. This is glucose. This is galactose. Galactose. Okay. What's the difference between that? And then galactose. What's the difference between that? It's in the carbon number 4. This hydroxy bond is go down, this guy is go up. So what is it looks like a lactose? Lactose happened is 1' and 4' carbons they connected. So what happened is H, there's O, and there is a H connected like this. That's a lactose. And if bacteria expressed beta-glycosidase, what will happen? This lactose will break down glucose and galactose. So I'm not going to draw again. Okay, basically you break down these. But at the same time, you need to know they have a side chain reaction. Will generate another very side chemical. What is that? It's the same thing. So we draw real quick, okay? We draw like that. Then we draw right here. Briefly. So we're not going to draw everything. What's the difference? Lactose is 1,4 glycosidic bonds between glucose and the galactose. This is a major procedure for galactosidase to hydrolyze it, become glucose and the galactose. And this is the major reaction. The side reaction, what happens is, instead of I have a 1,4 glycosidic bonds, I had is a 1,6 glycosidic bonds. And the others are the same. Okay? So if you're still confusing, you can draw them all together. These OH and H up and down, it doesn't matter. It's the same thing. H, H, OH. It's just a different chiral structure. Okay, H, CH2, OH. The key point is right here. So what is this guy? Lactose. Okay, now the lactose you need to come in. You need to come out inside of the cell membrane system. What the enzyme you need is permease. Permit, like you can come in. So what happened to the story is, if there is a lactose, they will generate a little bit of galactose. And they will generate a little bit of allolactose. And the allolactose will be reacted to the tetramer, they stop attached to the O, and then the transcription will be happens. And this allolactose, we have to say, the amount has to be high. You cannot only have a minimal amount, okay? So that's why you need a lactose to do it. So that's a story for lacto-operon on the right side. So Menard and Jacobowitz, they find of the lacto-operon, then they go to publish the paper, then get a Nobel Prize. But later on, people find a very interesting structure. The results. What happened? People do a research. Their research is very interesting. They grow a bacteria, let's say E. coli, in the co-present of glucose and the lactose. What happened? This is the time, x-axis, y-axis is the cell population. And they find the results is very interesting. There is a double growth curve for bacteria. At the beginning, they only use glucose. And later on, they start to use lactose when the glucose is gone. This is called dioxic growth. So what happened? They find in a bacterial medium, if there is a glucose and there is a lactose, the bacteria use glucose first. When the bacteria used up all the glucose, they started to use lactose to survive, have a second type of growth. So I give you a joke, is that in your home you have a lasagna and you have a bread. All the time you're eating a lasagna. And one day you find in the refrigerator the lasagna is gone, and you tell yourself, I'm going to go ahead to eat a bread, otherwise I'm going to be very hungry. That's what for the bacteria. Now how bacteria can do that? I remember we talk about the lacto-operon right now is PO. We did not mention SC yet. Is that right? So we're going to have to mention SC. So what people find is that for the lacto-operon to be really working, which is CPO, lac Z, lac Y, and lac A, not only they need to have lactose, they also have to make sure there is no glucose. Because once no glucose, they will activate CAMP, which is cyclic adenine monophosphate. This guy have to be attached to the CAMP. And here, you know, the tetramer is released from the O, then the RNA polymerase could attach to the P, then they started a transcription. So we need to talk about here then the story. How come the glucose connected to the CAMP? This needs to back to our knowledge from exam 1. Okay, where are we going to talk about that? Do you still remember the cell wall structure? You all know that. Fluid mosaic model. You never will forget. So we draw it. Phospholipids right here. Okay, We have a glucose, right? Okay. And remember, we talk about glucose become glucose 6-phosphate, then become glucose 1,6,5-phosphate. Then we go through this guy, glycolysis, generate pyruvate. And I said a very important chemical right here is called PEP, phosphoenolpyruvate. Do you remember, if there's a glucose comes for the bacterial cell, what they do? This PEP will be transferred, go to complex, enzyme complex 1. Then we'll go to HPR, heat stable protein. And then what they go? Remember, there is a bunch of stuff right here, which is called enzyme 2 complex. And then what happened? This phosphate goes here, this phosphate goes here, and then they go inside. The glucose becomes what? Glucose 6-phosphate. What is this called? Group translocation. And I said very clearly, glucose becomes glucose 6-phosphate for E. coli cell. If it's a bacteria, that is going through group translocation. Is right what happened right here. And in fungi, they rely on enzyme, hexanase. So this is what happens. There is a glucose. Okay, we draw a little bit long. We talk this guy out. Okay, we need to use this space to draw it. So this is what happened. There is a glucose. Okay, if no glucose, then what happened? If no glucose, which means this line will not be connected to go there. What the phosphates, what are going to be go? They're going to activate an enzyme. The enzyme called AC. What's that? Adenine, adenyl cyclase. What's adenyl cyclase for? They will let ATP becomes CAMP and release 2-phosphate. This is what the CAMP comes from. When there is no glucose, they go this direction. They don't go that direction. So what is this reaction for? What is ATP? Can you draw it? You should know how to draw. NH2, N, goes here, 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 here. This is H. This go here. Then you go here. This is a phosphate. Okay. Then you go H, 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 OH. Then you go here H, CH2. You have O. You have phosphate. O, phosphate. P, phosphate. We have O, P, like this. Okay. If you have a cyclase, what are you going to do? Once you have a cyclase, PPI will be gone. 2 phosphates will be gone. So these 2 are gone. what they becomes? Everything else stays the same. So we can draw one more time. Don't hesitate to draw it. It's very important you learn how to draw these things, but you will understand it. NH2, N, and you go here, which is carbon number 9, and you go here, H, OH originally, is that right? CH2. What happened? This is gone. This will become an O, become a P, disconnected. This is C, A, and P. And I make a joke all the time. This is very funny. This C and P comes, you look at the ATP, what's the difference? It's a circle. Once it makes a circle, what happens? Like the money is being bounded, you can't use it. From the energy standpoint, that's why all the disease, when it happens, you have a mucus come out, is ATP becomes CNP. Then what happens here? They generate CNP, so it goes to here. They attach. But here's a good thing. Okay, they goes to here. They go to CNP. CNP attached to the C. They start to do the transcription. Now how they do it? The CNP will actually combine with C position, the catabolic activated binding site. They will activate the alpha subunits of RNA polymerase. Then let the RNA polymerase to connect it to the P, to the promoter, and start to do the transcription. So that's what happened. Okay, therefore, this is a big story. Therefore, we know lacto-opera, at the end of the day, it is actually a combination of positive and negative control, which makes it complicated. Therefore, in the real life or in your other classes, there is a 4 different situation will be happen in the lifetime. And we will be explain this to you, these 4 scenarios. So I use this type of the blackboard, we explain this. What happened? I usually draw this structure. You don't hesitate to draw it, because you draw, you understand. Okay? 4 different situations. And everything here, we're right. Yes, CPO. Lac Z, lac Y, lac A. Same thing here. CPO. Lac Z, lac Y, lac A. CPO. Lac Z, lac Y, lac A. The last one, almost done. Little patient. CPO, lac Z, lac Y, lac A. So, we have glucose, we have lactose. Presence, absence, 2 by 2. Basic math question, how many different scenarios? 4. Let's talk about that. First situation, there is glucose, there is lactose. Okay, what's going to happen? First of all, there is a glucose there. So, CAMP will not attach to the C. There is a lactose there. Luckily, what is going to happen? This tetramer will be attached to that, allolactose, so will not attach to the O. However, because the camp is not attached to the catabolic site, not attached to C, so this AEM polymerase will not attach to the P. Therefore, the transcription happen or not happen? Not happen. This makes sense. There is a glucose, there is a lactose. So bacteria use glucose first. This tells you the scenario which is at the lower part of the figure. That makes sense. Okay, let's talk about second scenario. There is no glucose. However, there is, lactose. What happened? Because there's no glucose, so they did not generate glucose 6-phosphates. Instead of, they generate CAMP. So what happened? Can attach to the C site, and because there is a lactose, so the allolactose will be generated, and right here, and this is not attached to the O, therefore, RNA polymerase attached to the P position, attached to the promoter, the 5' to 3' transcription start to happen. That makes sense, is the higher parts of the figure. Okay, let's talk about something else. How about yes, there is a glucose. However, there is no lactose. Is the lacto-operon going to operate it? No, of course not. Because there is a glucose there. The camp will not generate, it's not going to attach to the C. And there is no lactose, so the tetramer will be stacked to the operator position to prevent the RNA polymerase attached to the promoter. Therefore, this 5' to 3' transcription not happen. That makes sense. If there is a glucose, there's no lactose, of course, bacteria are going to use glucose. You have a lasagna at home, there's no bread, of course, you have to eat lasagna, is that right? That makes sense. Okay, last situation. There is no glucose, and there is no lactose. What happened? Okay, no glucose, that's really nice. CAMP will be generated because it's activating the cyclase, so they connect it to the C. Because there is no lactose, the tetramer is going to be stacked to the O position. So what happened? The RNA polymerase will not attach to the promoter. Therefore, this 5' to 3' going to happen? No, of course. This also makes sense. You don't have anything. You don't have lasagna, you don't have bread, what you going to do? Go grocery store, buy something. Is that right? Eat some chocolates. You get chocolates yesterday a lot. Yeah, just get some chocolates. That makes sense. There ain't nothing going to happen. What are you going to have to do? Go grocery store. The bacteria need to find some alternative energy resource. Maybe maltose, maybe sucrose. That's what we talk about the fermentation. That's what it is for. Central metabolic pathway doesn't work. Go fermentation. Yeah, that's what it is. So, what happened is there's a four different situations could well explain what it is. The story behind this is a little complicated. If you understand it, you go ahead to do, but I would actually, in the exam, you should know the results of these. You can fill the table, the 4 different situations. Okay? And we will review again on Tuesday. To remind you again, this will be tested because it's a basic knowledge. And I understand it's like a maze. For some of you, first time learner, but you already learned in your other classes. Here we just focus on, again, to talk about that. But when we talk about in this class, because originally this did on the E. coli, and they are coming from the study based on glucose and lactose in a dual nutrient medium to see the growth situation of E. coli. You have a double growth curve. And it's a fit for general microbiology. That's why we want to point it out to mention this again. And also, this is connected to the knowledge of what we learned before about exam 1, which is group translocation. That's why I always say the glycolysis at the beginning of the stage is important because the difference between eukaryotes and the prokaryotes is at the beginning. One is using hexokinase, another one is using group translocation. Okay, that's what we have. I don't have enough time to finish talk about the others, so this is a picture, well explained, what I draw on the blackboard. If you really don't like it, this is a cartoon movie picture for you to understand easy. If low glucose, then adenyl cyclase will be activated and become CAMP. CAMP attached to the cap starts the transcription. And this is exactly what I talk about right here. The 4 situation. This is exactly what I talk about. I just have a slide to show you. Okay? I don't have enough time, so the last section I'll just skip it. You learned in your other classes, restriction enzyme, sticky ends, and the last one, just be very briefly. It won't be in the exam. Just tell you, people have these vectors. They know lag Z, lag Y, lag A finally will be expressed. So they're going to use lactose or not. Based on use lactose or not, they have those restriction enzymes to cut those vector sections and do the recombination, and finally they could be verified in a bacterial media based on the color change. It's a very smart way. It's a traditional spread plating methods with modern molecular methods. Well, not really modern anymore, but at the beginning, it's really nice. It's a 2 technology combined, which is the microbiology, biochemistry, and the genetics they combine. So that's why this is, if I have enough time, usually I talk about that. But start from here, we will be just skipping, okay? So, this section, important is this slide, to talk about the results of 4 different situations, 4 different scenarios, what it's going to be looks like, okay?